Okay, so now we're moving on to our adaptive immune system, which is our body's built in specific defense system or our third line of defense. It does work with an innate defense system, like we talked about before. But if we don't have a properly working adaptive immune system, or if it becomes disabled, we can result with some sort of disease like cancer or even AIDS. So activities, if we're talking about the adaptive immune system, it's going to help amplify that inflammatory response even more, and it's going to activate the complement, which we talked about both of those in the previous section. Downside to the adaptive defense, it has to be primed by initial exposure to foreign substance, and priming takes time, meaning it has to be exposed to something in order for it to be activated and ready to go. So some characteristics of the adaptive immunity is one, again, it's specific. That's the definition of the adaptive immunity, meaning that it recognizes and targets specific antigens. And those antigens are those things on the surface of cells. It's systemic, so that's a good thing. It's not restricted to the initial site of injury. Also, it has memory. So it can mount an even stronger attack to known antigens if we have another exposure or even more than one exposure. There's two main branches of the adaptive immune system. We have humoral and cellular immunity. If we're talking about humoral immunity, we're talking about using antibodies. If we're talking about cellular immunity, we're talking about using cells. Okay, so a little bit more about humoral immunity, which is also called antibody-mediated immunity. Is going to be provided by the antibodies that are present in our body fluids. They're produced by lymphocytes, and antibodies are constantly circulating around in our blood and our lymph. And they can bind primarily to extracellular targets, so things like bacteria, toxins, and even viruses. And when the antibodies bind, they can inactivate them temporarily. So they don't actually kill them, but pretty much they go in, and by binding an antibody to whatever it is, the toxin or the bacteria, you're marking it for destruction for a phagocyte to come in, or for it to undergo complement. We also have cellular immunity. So if we have lymphocytes rather than antibodies coming in to help protect the body against microbes, that's when we're talking about cellular immunity. So cellular immunity has cellular targets. So again, cells that are infected with pathogens like viruses or bacteria, even cancer cells, or any other foreign microbe. So the lymphocytes themselves are going to act against their target either directly by going in and killing the infected cell, or indirectly. We can go in and just release chemicals, and these chemicals are going to enhance that inflammatory response. They're going to help activate macrophages, or they could even help the entire adaptive immune response to understand what it's supposed to do and what's supposed to go on. So let's talk a little bit more about antigens. We've talked about them a little bit before, but just so we're kind of all on the same page. If we're talking about an antigen, that's a substance that can mobilize the adaptive defenses and provoke an immune response. So pretty much anything that's on the surface of a cell could be considered an antigen. Antigens are the target of all adaptive immune responses. Most antigens are really large. They're complex molecules that we don't normally find in our own body. And the characteristics of antigens is that they can either be what are considered complete or incomplete. If they're complete antigens, we call them complete antigens. If they're incomplete antigens, we call them haptens. Antigens contain what we call antigenic determinants and can be a self-antigen, which we'll talk about all of these now. So, like I mentioned, antigens can be complete or they can be incomplete, in which case they're considered a hapten. Complete antigens have two important functional properties. One is immunogenicity, and which is pretty much the ability to stimulate a specific lymphocyte to proliferate or multiply. And the other one is reactivity, which is the ability for it to react with the activated lymphocytes and the antibodies released during immunogenic reactions. So, for example, foreign proteins, polysaccharides, lipids, nucleic acids, those can be seen as foreign invaders, or even things like pollen or other microorganisms. Incomplete antigens or haptens involve molecules that are too small to be seen. So they're not really immunogenic by themselves. So one example is small peptides, nucleotides, and some of the hormones. However, they can become immunogenic and cause an immune response if we take these haptens and attach them to our own proteins. So then we get a combination of our own protein and a hapten, and that can be seen as foreign. Downside is if we have these haptens that go in and attach to our own proteins, that can cause our immune system to mount an attack against our own proteins because they have this hapten attached. So examples of when this happens, poison ivy. So poison ivy will go in and complex with one of our own proteins and mark it as foreign. So our own immune system goes in and attacks it. Same thing if we're talking about allergies to animal dander, detergents, or even cosmetics. So antigenic determinants. So only certain parts of the antigen, which we call antigenic determinants, are immunogenic, meaning they cause an immune response. So our antibodies or the lymphocyte receptors that bind to these determinants, kind of in the same way that an enzyme binds to a substrate. It's a very specific type of binding. Most naturally occurring antigens have not just one, but multiple antigenic determinants that can mobilize several different lymphocyte populations, as well as form different kinds of antibodies against them. So large, chemically simple molecules such as plastics, they typically have little or no immunogenicity. So here's an example. Here's our antigen right here in red, and we can see we have all a bunch of different antigenetic determinants. So here's one antigenetic determinant, another one, and another one right here. So all of these antigenic determinants on the presence of whatever antigen we're talking about, different antibodies can go in and bind the different antigenic determinants, but they are specific. So it's almost like putting a puzzle together, because again, we're in our adaptive immune system. So let's talk about self-antigens. So again, we're talking about things that allow our immune cells to recognize that, hey, it's our cell. Don't mark it for destruction. Don't mark it for as foreign, because we are displaying what we call self-antigens, assuming that our immune system is properly programmed. So your self-antigens are not foreign or antigenic to you, but to somebody else, they would strongly be antigenic. So this is why a lot of times there are rejections during transfusions as well as grafts. So on the cell surface, some of the cell surface proteins that identify a cell as self is a group of glycoproteins that we call MHC proteins, which I think I talked about earlier. MHC just stands for major histocompatibility complex. And coded by genes of the MHC and unique to each individual. So they contain a group that can hold a piece of the self-antigen or a foreign antigen. So if we're talking about T lymphocytes, they can recognize only antigens that are presented on MHC proteins themselves.